Now this is all nice if you know exactly the composition of the genome. And of course, all the biological experiments are noisy, and so there are always errors in these reads, and some reads are, like we said in the initial New York Times newspaper exploding experiment, some of the reads are lost, right? And so basically then how do you handle such errors? And there is also this issue that I told you, that if k is very small, then you will have two genomes which will have the same composition. So how do you handle all of these issues? And that is where the technology of read pairs come in. Okay, the read pairs, in order to motivate it, let's just go through the summary again. From the composition, you construct the de Bruijn graph, and there you construct the Eulerian circle to get the genome. Okay, that's what our current approach is. And given a particular de Bruijn graph, like I said, there is no unique genome. So, right, I mean, and in this particular example itself, we can show two different genomes that will have the that will basically have same compositions, right, and also this therefore the same Eulerian graphs, right, but different paths. There may be more than one Eulerian path in the graph. So, in each Eulerian path will correspond to one distinct genome. So here, for instance, this is our genome, and then this corresponds to a certain path, and then here you can see that there is a difference here. So it went by a bit fast. So let me just do it again. So it is taking up this up first and then down, but here, and then you get this particular string. Okay, so let me just point out the key difference. So these edges are visited first before these edges. These are visited second. Okay, in this particular formulation. Okay, so I will show you again. So you're going through the center, then you're going up, then you're coming down, then you're going right. Okay, and then you get this particular genome. But you can also go down first and then up second, right? So then if you do that, then this GGG is going to come before whatever is here, right? GCC, right? So here GCC came before GGG because GCC is over here, GGG is over here, right? But if you focus this first, if you visit here first and visit here second, then you in your genome GGG will come before GCC, right? So you can see here that it goes into the center, it goes down, it goes up, and then it goes to the right. So you can see here that GGG has come before GCC, right? So these are two different genomes which have the exact same composition, right? You can check that, and then and they are just so they will have the exact same de Bruijn graph from that composition, and then they are just different paths, different ordering of the way in which you visit the edges. Okay. So one technology that allows you to get around this is read pair sequencing. Okay. So what does read pair sequencing mean? So like I have told you before, the sequencing technology you can only look at reads that are short in length, right? But then if you want to increase the length of reads without actually improving the technology a whole lot, you can look at a fragment which is a piece of DNA, and let's say you can only measure 100 letters at a time, 100 ACGT, 100 nucleotides at a time. This may be 1000 in length. Okay. Instead of breaking it into 100 and then getting each of those 100, you can just do at one end 100 and another end 100, and this will be called read 1 and this will be called read 2. So these are called paired reads. So then you have read 1 paired with read 2. This is 100, this is 100. Okay, what will this be then? 800. Right? This 800 letters we are not reading, but the 100 letters at the end we are reading. Okay. The no technology, let's say technology cannot do 1000 letters at a time, but the technology can do 100 letters on one end and 100 letters on the other end. Okay, so that way you can get a little bit more information, and you will be able to tease apart these kinds of issues. That let's say you have a longer read that has GCC and GGG, right? So you have GCC, ATG, GG, right? GCC, ATG, GG. So you may have this as your long read. Let's say you can only do 3 mers at a time, but if you can do in both ends, then you will know that GCC is here and GGG is here. So you'll know that this is read 1 and this is read 2, and so GCC has to be to the left of GGG. You can figure out. rather than this where GGG is to the left of GCC, right? Without actually learning what is in between, right? So you will just have GCC dot dot, you will have GGG, right? So you will learn this and this as read 1 and read 2. The dot you won't really read because the sequencing machine cannot do more than 3 letters at a time, let's say. But you can still figure out the relative position of this GCC and GGG. Okay, that is the idea in this read pair technology. Okay. So the idea is that you take multiple identical copies of the genome like before, you randomly cut them into now larger equally sized fragments. Previously we had to split it into k-mers, but now we can also make it like 10 times k-mers or 20 times k-mers. You can make larger fragments now, right? But then you still read k-mers, but at the end of each of these fragments, right? So if you have this is called the insert length. You have 200 base pairs at each end, and then you are just measuring this, okay? So you generate read pairs, 2 reads from the ends of each fragment separated by a fixed distance to get around this GCC GGG problem that we just saw, okay? And this is an interesting exercise now on how will you build the de Bruijn graph with these paired reads, okay? In the sense that with single reads, we could take each read and split it into these k minus 1 mers and attach those k minus 1 mers. But with pairs of reads, how will you do it? And that is what we will see in the next set of slides, okay? So you have a read 1 and you have a read 2, and this is the insert length or a distance of d equal to 11, okay? A paired k-mer is a pair of k-mers at a fixed distance d apart in the genome. So the sequencing technology allows us to do this. So here are TCA and TCC. The k is 3, but the distance d is 11. Okay, so they are 11 distance apart. But then the sequencer has to only read 3 letters at a time. Okay. I mean, there are, of course, in reality, read 1 and read 2 are typically sampled from different strands. Here we are just showing it as reads from the same strand of the DNA. And also in reality, the distance d is measured with errors, right? It's not always exactly 11 apart, but in the spirit of this course, we always kind of make the problem more cleaner. We look at a toy version of the problem. We address that problem first, design algorithms, then we add some bells and whistles to the algorithm so that it can handle real world data, which is much more messy than the clean problems that we see in the class. Okay. If I start showing all these messiness, then I will lose most of you. So okay. So what is paired composition? So before we even go to paired de Bruijn graph, we have to know what a paired k-mer composition is, right? So a paired 3-mer is essentially this KDK triplet, right? So you have a paired 3-mer, here is TAA-GCC, right? So this is like a D equal to 4 here, right? So here D was 11, right? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, right? So 11 was the distance here. So here D would be 1. So TAA, and then there is a 1 gap, and then GCC, right? And these are all your paired 3-mers with with an insert length of 1 in between. This is again, there may be some repetitions here. And so this is a bag. It's not a set, it's a multi-set of these k-mers. Okay, you can allow some of these paired k-mers to be repeated. And just for simplicity, it may be easier to just represent it as a 2-line expression. So just to reformat it so that it's easier rather than these kinds of writing it in the single line. So this TAA GCC is going to be written as TAA and then GCC below it. Okay. Now you can think about a de Bruijn graph that is exactly the same as before. So de Bruijn graph, if you recall, you put edges and then on the edges you put k-mers. Here also you can put edges, on the edges you can put now paired k-mers, right? And then on the nodes you can put paired k minus 1 mers. Okay, that is all the difference that is required. So you can again sort it out in a lexicographic order because we do not know the original position in the genome, and then you can frame the problem. You can again make sure that you frame the correct computational problem. You want to reconstruct a string from its paired k-mers. The input is a collection of paired k-mers, and the output is a string text such that the paired composition of the text is same as the paired composition of the genome, right? And that is exactly stated here, like we have just said. And how do we construct the de Bruijn? Like I said, we just pad the de Bruijn. So we just take 2 copies of de Bruijn, right? And essentially we put edges where the edges are labeled from these paired k-mers, okay? And then the nodes are essentially these prefixes of both of them. So you can see here that you have the CC and GG is shown here, and then the suffix CA and GG is shown in the next node. Okay, and then you split each of your pad k-mers into these two nodes and an edge, and then you start gluing all those nodes together like we did before. Okay.
Like, what's the advantage of having this paired k-mer? Like, read pair is something, but
yes,
this paired k-mer, like, what's the advantage?
The pad k-mers is essentially the way of utilizing the read pair information to avoid these ambiguities in the genome reconstruction. We showed an example where GCC was before GGG in one reconstruction of the genome, and then the other one GGG was before GCC. If you have pad k-mers, or if you have pad reads, you will be able to resolve that ambiguity and say that only one of the genomes is valid. Okay, but in order to solve that problem, we need to have an Eulerian cycle algorithm that will handle these pad reads, right? And so pad k-mers is a way of designing a data structure and an algorithm that will handle paired data. Right. So what I'm trying to say is that there is ambiguity. So this is our resolution. There is ambiguities. Oh yeah, because you are doing in this experimental process, like we said, every genome is duplicated multiple times.
Okay, so
using the PCR or other experimental processes,
so, like,
right?
Different composition will be there.
Exactly, yeah. Because this random shearing will happen at different places in the different copies, right? Exactly, yeah. So because each one of them, this is copy 1, copy 2, copy 3, copy 4, and then some of them here it may split here, but in another case it may split here, right? In another case it may split here, and so on. So for one genome, at every possible pairing you are assuming you are able to get if there are enough number of these copies. Again, in real world you may not get every possible pad k-mer, but you are assuming that if there are millions of copies of these genomes and then everything gets randomly sheared, then all possible pad k-mers are output by the sequencing machine. Okay, so we have ambiguities, like one kind of k-mer composition can give you multiple genomes, genome 1 and genome 2. Though this cannot be fully resolved by pad k-mers, if you look at pad k-mer composition, you may be able to get only a unique genome. Okay, more likely to get a unique genome because some of the ambiguities are resolved. But to go from a pad k-mer composition to get a unique genome, you have to go through the pad de Bruijn graph, and then you need to find an Eulerian cycle in the pad de Bruijn graph or Eulerian path to get the unique genome. Okay, that is the strategy. And to first do that, we need to construct this pad. So this is step 1 and this is step 2, right? So the step 1 is what is shown here. So we have each of these pad k-mers are written on the edges, and then we have pad prefix of and the pad suffix are the nodes, right? And then we put it in, we then go ahead and glue together all of them. So there is a TG80 and a TG80 here, and we glue them together. And this is the pad de Bruijn graph, and you can see here that because now there is TG and also an AT, right? So there is not, previously we had 3 nodes that we had to glue, but now there is only 2 nodes that we have to glue because this extra pad and information, pad read information, will minimize the number of duplicate nodes in this. And if you minimize the number of duplicate nodes, then there is also a lesser number of choices you have to make and lesser number of genomes that correspond to this de Bruijn graph. Okay, and you can do the same. So instead of from the genome, you can do it same from a list of k-mers also, right? Which is what we do not know what the genomes are. So from a list of k-mers, we can look at all the pad k-mers that we have. Each one of them we can split it into this suffix and prefix, right? Pad suffix and pad prefix. Then we can start gluing them together, right? All those with identical labels. Okay, it's exactly the same process as before, but instead of one row here, now we have 2 rows. So this is row 1, this is row 2. Okay, and that means that there is lesser gluing to do. If there is lesser gluing to do, then there is more likely of getting a unique genome. Okay, lesser ambiguity in the genome reconstruction. Okay, so the pad de Bruijn graph is you represent every pad k-mer as an edge between its prefix and suffix, and you glue all the nodes with the identical labels. So here is some gluing that happens. This is the first level gluing, and then the second level gluing is actually minimized if you use paired k-mers versus normal k-mers. There's only one gluing here we have to do, and then this admits only one Eulerian cycle, right? So you have to go through like this, and then you go down like this, and then you come back like this and go like this. So this is the start, this is the stop, and that will give you the exact genome. Okay, this is something that you have already said. And just to show again how paired de Bruijn graph is better than a de Bruijn graph, like I said, there are many loops here, many choices here. You can go up here and then down here, or you can go down and then up. That kind of ambiguity is resolved here. There's only one way to go here. There is no two Eulerian cycles. There's only or one Eulerian path. There's only one Eulerian path on the left side paired de Bruijn graph. Okay. So this gives you a unique genome reconstruction. The right one gives you multiple genome reconstructions. So the final the part of this whole session is about de Bruijn graphs are great, and along with read pairs, it gives you a unique genome reconstruction. But still, there are many details that we really pushed under the rug because we, like you just raised, you don't get all the paired k-mers from a sequence. Some of them may be lost, right? And then some of them may be error prone. Like you may think that one of the reads is TAA GCC, but what if this A is actually a C? That this is actually TAC and GCC, right? And there may be some sequencing errors. So what will happen in terms of errors? Yes, we may not get enough amplifications. And then what if the points where they are being randomly shared or not exactly random? So there are all those harsh realities. And so de Bruijn graphs, how do they face the harsh realities of assembly? Usually the way that these will go is you construct a clean algorithm based on a clean problem. Then you add some additional heuristics to the algorithm so that they can get around some of these issues. Okay, we won't get into it in very much detail, but we'll just briefly mention some of these. Okay. And some of these assumptions are so ridiculous, right? So they are in the sense that all of the sequencing instruments are error prone. They're highly noisy, right? So we assume that reads are error free, right? So that is one big assumption. And then also there's another big assumption is we assume that there are millions of copies of the genome and all possible random cuts are possible. And that there is a perfect coverage of the genome by reads. Like every k-mer from the genome is represented by a read. That is also not the case, right? Some of the k-mers may be represented more, and then some of them may be represented less. And the multiplicities, the number of times a k-mer duplicates, the number of copies of a k-mer also was known. So, for instance, we said that ATG was present 3 times. This we may not know in exact experiments, right? We may just know that ATG is present. We may not know exactly how many times it is present in the genome. And the distances between reads within a read pair are exact. Again, this is just a distribution around some distance and not always precisely some simple small number. Okay. So the coverage is imperfect, the reads are error prone, and the multiplicities are unknown, distances are inexact, and then so many other things. Like I said, we won't try to address all of them here, but we will try to see if some of them, how you will approach addressing them, right? So the first unrealistic assumption about perfect coverage, right? So there is this 250 nucleotide reads generated by this Illumina technology, right? And then, but they only capture only a small fraction of all the 250 mers from the genome. So if you have a genome which is 3 billion letters, and there are like multiple one of these 250s, right, that this Illumina covers, right? But there may be some regions of the genome where it doesn't cover at all, right? Or it may only cover very less. It may not cover perfectly, right? Every possible k-mer from that region may not be possible. So it violates one of the key assumptions of the de Bruijn graph. So how will you fix this, right? So with k equal to 250, we are saying that it's not going to give you all the 250 mers in the genome. What can you do instead? Any guesses? Increase k a bit from the experimental side, you are saying. Okay, so you want to do k equal to 500 if the experiment allows it, and then operate it computationally at 500 mers, or 500 mers may have more of this issue, right? Because 250 mers itself, you are not able to cover all 250 mers in the genome. If you do a sequencing with 500 mers, then it may be even more problematic, that even more sparse coverage of the genome. But the answer is along the similar spirit, but it's not increasing. What's the other option? You just decrease k, right? So essentially, you just you can see here that this particular k-mer is not covered, for instance, right? So this from here to 1 2 3 4 5 6 7 8 9 10. Yeah, this is 10. Okay, so 1 2 3 4 5 6 7 8 9 10. Okay, till here. Okay, so from here till here, this is not covered if you use k equal to 10. Okay, but let's say that I split it and I use k equal to 3, then each of these will split into multiple 3 mers, right? I mean, there are also overlapping 3 mers. And so then almost all of the 3 mers in this string will be covered if you reduce k. Okay. So that is the what is illustrated here. So you want to break your reach from longer k mers to shorter k mers. Okay. So here they are 10 mers, but now you are making them 5 mers. If you make them 5 mers, then you are able to cover every location in the genome, all the k mers that start at every location in the genome, like shown here. Okay. So all this will cover all these locations, right? And then this will cover these kinds of locations, and similarly, right? So these two additionally will cover from the blue read, and then these will come from this I don't know the color, violet, I guess, and then this will be the green. Okay, so then you are able to cover all the k-mers for a shorter k, smaller k, by breaking reads into shorter k-mers. Then you can construct the de Bruijn graph using the smaller k-mers. But what is the disadvantage of smaller k? Like, why not use k equal to 1? And that is the extreme, right? Yeah.
Like they will never have the order.
Yes, you will never have the order. There's just too much ambiguity, right? So if you use k equal to 1, right, then ACGG genome will also have the same one mer as
Like node and age,
AGGC.
everything will be separate, but start and stop.
Yes, yes, exactly. Yeah. All of these are, there are many ways of ACGG. All of them will have the one mer composition, 1A, 1C, and 2 Gs. Right? This is your k-mer composition. All of these genomes will have the same k-mer composition for k equal to 1. So as you make k smaller and smaller, there is more and more ambiguity on what is the correct genome. Many genomes will be possible for the same k-mer composition. So we want k to be as large as possible, but the technology limits having k to be very large. So you can start with, let's say, 250 mers that Illumina gives, then maybe you can convert it into 25 mers. And then try to process your de Bruijn graph with 25 mers. 25 is still a reasonably large number that you won't get this ambiguity issue in genome reconstruction. Okay. So the second unrealistic assumption was error-free reads, right? And then this one is interesting because the de Bruijn graph starts getting, if you have errors, starts getting some kind of weird features it develops. Okay, so I'll just show you what it is. So let's say that this T is converted into erroneously into a C. Okay, there is some sequencing error, and then the T becomes a C. Okay, so then what happens is that in all of these cases, right? So you will have a CGTAC, GTACG, and so on, right? So these are all the k-mers, 5 mers, right? That gets affected by the sequencing error of this at this location, right? There are 5 such k-mers that will get affected by the sequencing error. So in addition to the original k-mers, these are some new errors that are being introduced. So if this is your de Bruijn graph constructed from error-free reads, okay, now this kind of errors will create something called a bubble. This is what I said, like the de Bruijn graph will develop some extra features. This is the bubble feature that it develops. That in addition to the original path through the genome, if there are some larger k-mers which cover this path, these bubbles will cover the smaller paths. Okay, these erroneous things, and then it will look something like this, right? So this may be the original genome, and then around every path there will be lots of bubbles around it, and each bubble will corresponding to a sequencing error. Okay. And then now you have some heuristics which you can add to remove bubbles, right? So you look at bubbles as, okay, you have some main path, and then you have some secondary path that connects two nodes along the main path. So is there a way to then differentiate between main paths and auxiliary paths, and then remove those auxiliary paths as sequencing errors, right? And because they'll especially have this kind of C being repeated everywhere, right? So can you do that and clean up your bubbles? And this is your one of the smaller organisms de Bruijn graph. Okay, humans will have millions and millions of nodes. There's smaller ones, and this is, you can remove the bubbles, and this is how things are after removing bubbles. Okay, yeah. So you can try to remove all of these little bubbles, and then you'll be able to get something like this. Okay, And so we have seen some of the problems like error-prone reads, how you can handle by bubble removal, and then coverage you can handle by reducing the length of K, breaking larger k-mers into shorter k-mers. So now all of these are done in real world to get the sequences of different genomes. And now we come to the question we posed in the beginning: who are these people? Any guesses? Yeah. These are the people whose genome has been sequenced in this personal genome project. So you have PGP1. So several others who basically contributed their genomes, and they basically wanted to sequence their genomes. This is a personal genome project. 10 scientists and entrepreneurs who made their genomes available in 2009. Right?
Francis Collins and Craig Venter did the
No, they didn't want to reveal that for privacy reasons. They didn't want to reveal, but there are
Yeah,
certain people who
sequence
said, "I don't have concerns about privacy. Here is my genome. Here is it. Here it is." And we had this question in this molecular biology glossary also. We have as reference human genome, which was done. Um, whose genome is it? Do you know? Whose genome is the human reference genome? Whose genome, meaning that each one of us, we just said that in 3 million letters we are different. Like 1 in every 1,000 letters we are different. But then a human reference genome has some set of 3 billion letters, right? So whose genome could it be?
Watson. I'm just guessing.
Uh, yes. It's actually we do not know whose it is. There were some people who volunteered their DNA for the Human Genome Project, and it's most likely the majority allele of those different people. Yeah, the majority.
Like statistically significant.
Yeah, each one you just look at what is the one that is present in most of the individuals, and then you look at that. For places where the genomes differ. But for many places, except for the 3 million, rest of the 3 billion are all mostly similar across individuals. They are all the same. So just wanted to end with the note that because of all these technologies, in terms of both experimental technology and this graph technology, this graph algorithm that is at the heart, Eulerian graph algorithm, and several other algorithms, there are also heuristics to solve the Hamiltonian version of the problem. But mostly these types of different algorithms that makes large scale sequencing possible, assembly possible, we are able to sequence lots and lots of these species as well as lots and lots of disease genomes as well. So, okay, with that, I will end this session. Thanks for your attention and interest.